Howdy and welcome to the unit on primary structure, the protein sequence. This bullet list tells us the features of primary structure that are true for all proteins. The primary structure or sequence is unique for every protein, and it is encoded by the nucleic acid sequence of DNA. So there's information in this biopolymer. It's read from the amino terminus or N terminus to the carboxy terminus or C terminus, which means that the convention is N terminus on the left, C terminus on the right. So like our nucleic acids, our polypeptides have different ends. They have information and they have unique beginnings and endings. All of the information that's needed to fold a protein into its correct three-dimensional structure and therefore attribute its function are actually encoded in the primary structure. That is, the sequence from N to C terminus instructs protein folding and function. This is a video that you can watch as a study break. Part 1. There is a limited diversity of polypeptide sequences. Now, this isn't necessarily expected for these biopolymers because you can imagine that even a small polypeptide, like 100 amino acid residues long, would still be 20 to the 100 possible different sequences. 100 amino acids is very short. It's a small protein. Most proteins are much bigger. So in this example, 20 amino acids to the 100 possible different sequences is 1.27 times 10 to the 130 possibilities, more possibilities than the estimated number of atoms in the universe. So obviously that something is limiting the number of polypeptide sequences. So what's behind the constraints on what polypeptide sequence actually forms a good protein? A few of those are listed here in red. There's a limit to the efficiency of protein synthesis. It turns out that the protein machinery, the ribosome translating the polypeptide, the engagement of the tRNAs, the length and stability of the messenger RNA, all of those things are limited. And so the efficiency of protein synthesis is smaller than or equal to about 10,000 amino acid residues, although there is one notable exception we'll see. More importantly, the ability of the polypeptide to fold into a functional structure constrains the number of polypeptide sequences. For one thing, the lower limit of number of amino acids is about 40 amino acid residues. Any fewer than that, and you're not going to fold into a stable structure. But there are other things that constrain the ability of a polypeptide to fold into a functional structure, and we'll address those constraints more as we progress into the next unit. Suffice it to say that not all sequences will actually fold into a unique structure with a unique function. All right, so the fundamental structural pattern that you find in proteins is dictated by the repeat of atoms in the polypeptide unit, and that's called the backbone. And the backbone is shown here. We're talking about an unbranched polymer from N to C terminus with no branches coming off. So we're going to be connecting those amino acids one to the next to the next. And we talked a little bit about this in the amino acids section. Here we're going to expand on that backbone formation a little bit more. So the repeating unit of the backbone is nitrogen, the alpha carbon, and the carbonyl carbon. So the carbonyl carbon is CO for that carbonyl oxygen you're imagining coming up out of it. N, C alpha, CO, N, C alpha, CO, N, C alpha, CO. Okay, so that's the repeat that you're going to see for the atoms of the backbone. All polypeptides have the same backbone repeat. And the backbone repeat is formed in a condensation reaction. We've seen this now a few times, and here it is again with specific amino acids. This amino acid has side chain R1, and the next amino acid in the sequence has R2 as its side chain. That makes this the alpha carbon here and here. And this is the carbonyl carbon here and here. And then the N is the alpha amino group in the free amino acids, has the nitrogen. And in the condensation reaction, of course, we're going to be forming the amide linkage, that pink bond between the carbonyl carbon of amino acid 1 and the alpha amino group of amino acid 2, releasing the elements of water as a water molecule. That's the condensation reaction that we learned about at the very beginning of the semester. So we lose a water molecule and we form a dipeptide bond. Formation of a bond is expensive, and we pay for that with phosphoanhydride bond energy on the ribosome, where GTP pays for the formation of each of the dipeptides. You might remember that from when we were talking about nucleotides. All right, so because the condensation reaction removes water, what's left is a residue. And the residue is residue 1, amino acid 1, connected to residue number 2, amino acid number 2. So when we talk about the amino acids in a protein, we sometimes use the shorthand residue, just as we did for the nucleic acid residues. Let's zoom in to the structure of the peptide bond. The peptide bond is an amide linkage. It adopts the transconformation and holds that conformation. Let's look back again at the transconformation. I know we reviewed it before, but the carbonyl oxygen is always opposite that new bond, that peptide bond, from the amide hydrogen. That's trans. The reason that it's held in the transconformation is because that bond actually has partial double bond character, about 40% of a true double bond character. That leaves the amide group partially positive and the carbonyl oxygen partially negative. And those partial positive and partial negative charges, those permanent dipoles that we talked about in the first unit, are excellent hydrogen bond donors and acceptors. So think hydrogen bonds. Now the length of the actual amide bond, that new peptide bond formed in the condensation reaction, is shorter than a single bond, but because it's only about 40% of the double bond character, it is longer than a double bond. And all of that is due to the resonance structures available to that amide linkage, holding it in the trans conformation. Because of that partial double bond character and the trans conformation, there are 6 atoms in the peptide bond group that make up a plane, which is defined the amide plane. See how it's in red? So you're going to have to remember which 6 atoms make up the amide plane, and importantly, how that planar structure constrains a polypeptide as we start to fold it into higher hierarchy of folding functional units. Okay, so this is what the polypeptide bond looks like in a sort of perspective geometry. So what we're looking at is the R group is going away from us over here, and the H is coming toward us, and that's shown by the wedge shape of their bonds. Same over here, the opposite. Here's the carbonyl oxygen, and the amide group is here, and they're part of this plane. The other members of the plane are the alpha carbon connected to the carbonyl carbon, and the alpha carbon connected to the amide group, the amide nitrogen group. And so that's 6. 1, 2, 3, 4, 5, 6. And when we look at how the electron clouds actually form around that plane, we have this schematic shown over here. So here's our plane with the 6 atoms, just shown as a flat structure, a planar structure, right? And here it's tilted with the same 6 atoms, but you're looking at those electron clouds if this was a pure single bond. If it were a double bond between the carbon and the nitrogen, the carbonyl carbon, this carbon here, CO, and the nitrogen, then the double bond would be here, and the oxygen's electron clouds would look like this pi structure. But because we have a partial double bond, we actually have an electron cloud that looks more spread out like this. And for that reason, there's partial double bond character between our CO and our N group, leaving a permanent partial negative charge on the oxygen and a permanent partial positive charge on the nitrogen, making them excellent hydrogen bond donors and acceptors. This is the reason why these 6 atoms remain in a plane and the peptide bond is always trans. Okay, and this is just another depiction of that plane set on top of our perspective drawing of our polypeptide backbone. So here's our backbone repeat. C alpha, CO, N, C alpha, and then it would go on. And then the 6 atoms in the plane are the C alpha, CO, the oxygen, N, the hydrogen, and the next C alpha. All right, that's the end of this video. See you in the next one.